This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode number 331, recorded on April 17th, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from St. Louis, Missouri, Petra Levin.
It's great to be here. It's finally spring.
It really is. I don't know if it is here yet. It's a little chilly. Let's check out the weather. Here in New York City, it is currently 16 Celsius. It's not bad. I think 20 is when I consider spring. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone, and we're in Chamber of Commerce weather. It's a beautiful, sunny day here in Charleston, no humidity, and 73 degrees Fahrenheit. So it's quite lovely.
And we have our guest today from Tacoma, Washington, Mark Martin.
Hi, everyone.
Welcome.
It's currently 58 degrees Fahrenheit out here.
That's not bad.
No, it's nice and sunny.
This morning I had to wear a wool cap and gloves. That's how chilly it was. It was like 5 degrees Celsius.
Ooh.
It's just a little bit too chilly. I have to wait on the train station, you know? So.
Yeah, and the wind blows down those tracks.
The wind and all that. Okay, folks. Today we have, as usual, a snippet and a paper. We're going to start with a snippet from Mark. It looks like you just accumulate more stuff in the background. Is that possible?
You don't see the rest of it, so don't worry.
It's a lot of stuff. You know, you have a plant even.
I have lots of plants.
And that, I just saw it. The phage that's right behind you.
That
you gave me one. You sent me one of those.
I did. I've given several of those away.
And it's at home because you have another one that you sent me from your former student. She knitted it, right?
Yes.
Iris Rosenberg, is that her name?
Oh, no. That's... Oh, my goodness. That's Ruth Eisenberg.
Ruth Eisenberg. It was close.
Yeah.
Okay. She knitted this. It's up on the shelf. You can't see it.
Oh, yeah. Yeah. It should have your name.
No, it doesn't have my name on it.
Oh, well.
No, but it's fine. It's really pretty and it stands up on its own. But I don't know. It didn't make the bottom shelf. It's too tall. That's the problem. I can't put it on the bottom shelf. Anyway, Mark, you could talk to us about the China space station. Is that right?
I will actually do that. And the first thing I need to do, because even though Michelle's not here, I'm well aware that she wants me to do this in the right order. First off, this just came out last month, and it is a research article in the International Journal of Systematic and Evolutionary Microbiology. Its title is Nyalia tiangangensis, isolated from the China space station. Now, this is by Yuan, Sang, Dang, Song, Yin, He, Su, Guo, and Yin. And I am very sorry for any mispronunciations. I will keep this to a snippet because I really want to hear about Petra's paper. But the first thing that I want to say to everybody is, yes, the Chinese have a space station. It's been up since 2021. Its name in Chinese, which I won't pronounce, is Heavenly Palace, which is a great name for it.
That's cool.
It has about 340 square meters of space. It's been pretty much continually occupied. They do a series of different types of experiments up there, including not only people, but also plants. They were working a little bit with rice and Arabidopsis up there. This will come back in a second. And they were curious what would happen to the microbes up there. It's in a slightly higher orbit than the ISS. And I would argue that its occupancy isn't quite as much. So
maybe is it strictly Chinese scientists or is it international?
As far as I know, I don't believe, I can go look that up later, but as far as I know, it has been Chinese nationals.
Okay.
And the other thing our listeners may be curious about, 300 square meters is about the size of a 3,200 square foot house. So your average suburban house.
That sounds decent for a space station.
Well, the pictures I saw made it look a little bit cramped. But then again, tons is tons, as the saying goes when it comes to lift weight. It's assembled out of 3 different modules. They're thinking about adding a 4th and a 5th. We'll see if they do. Remember, the ISS is coming down in a few years.
Is that right?
Yeah.
Oh, yeah. Its orbit is decaying and they've run out of gas, so to speak.
So is this going to be like a controlled deorbit or something?
It's not going to be like Skylab.
Yeah, Skylab.
You want it to be under control, and there's that place in the South Pacific. I don't know the name of it. I call it Cthulhu's Lair for you, Vincent, but there's nothing there as far as like people, and that's where they like to have a controlled.
This is a big thing, the ISS.
Oh yeah.
I mean, are they going to take pieces off initially, like the solar panels and all that, or just going to let it plunge?
My understanding is on that, and I'm glad you asked because I'm a big propeller head on this stuff, is I believe that they have, do you know what Hall effect boosters are?
No.
So those are ion thrusters, and they can plug and play on those things.
I see.
Power them, and like the Chinese have it on theirs. It helps with attitude control. And so when you could boost it to a higher orbit. We could do that with the ISS, but the decision was made not to do that because it's old technology, and there will be plenty of private space stations going up pretty soon is the idea.
I hope none of Elon Musk's.
I just want, this is as close as I get to politics today, but I would like Mr. Musk to just stick with rocketry. That's what I'm hoping for. But in any event.
You mean get out of the U.S. budget? That would be great. I would love that.
I still am fascinated by just the Webb. What we can do with the Webb telescope is still.
The Webb is great. Webb is
they
amazing.
found that planet yesterday
with potentially life on it. A future TWiM, perhaps.
TWiM.
It's got dimethyl sulfoxide. That's right. And dimethyl sulfide both, they say. And then a bunch of people are poo-pooing that. But it's been seen multiple times, but it's one of those.
At least we can use it as a solvent. I mean,
Yeah, it's true.
Isn't that made by E-Hux?
Yes.
DMS,
yes, exactly right. Yeah.
Maybe there's E-Hux on the planet. Who knows?
Well, we've been through this with Mars. We've been through this with almost anywhere you look. When you see any of the biosignatures for life, everyone comes with abiogenic sources of them.
Yeah.
And I'm to the point where, if there's a way to go there and look, that's the only way we're going to solve it.
But 120 light years away is a long ride.
It is for a place that's not exactly in the happy zone around that potential star. It's a little bit smaller than Neptune, the extrasolar planet you're talking about. So it's thought to be one of those super Earths that has huge atmosphere, and so a lot of hydrogen.
But it is a planet.
It is a planet.
So what is it rotating around? Anything?
A red dwarf. And the red dwarf is its sun. So it doesn't have the blue sun or not the blue sun, the yellow sun like we have.
Got it.
Well, that was fun to talk about. I love the space stuff. But yes, Petra, I have a bet, by the way, with Ken Nielsen. He doesn't think there's any life on Mars, but I think it'll be found. Subsurface. All right.
What's going to be found by who? You think the U.S. is going to go up there?
I want to go there, Vincent.
Well, you can't. You're too old.
Yes, that's correct. I am.
So
why don't we just get a little thing to come and dig and bring it back? I mean, I feel like we could get better samples on manned too.
And they're going to try that. I'm a little worried about it. I've seen too many bad science fiction movies, but I like the idea of bringing a sample back because all of the techniques we've used in situ, don't get me started, I don't think they're designed for finding things adapted to Mars. For example, dumping dirt into nutrient broth that was one of the Viking experiments. I'm sorry. Anyway, thanks for indulging me on this episode. So the idea of what takes place because of human habitation is an interesting one. We already know, for example, that microbes can evolve under laboratory conditions. Why wouldn't they under conditions of microgravity? I would further argue that the environment there is more extreme than we give it credit for, not simply because of microgravity, but because of its quite a bit of difference in terms of things like atmospheric composition, because I don't believe they use the typical mix that we use or that we enjoy on surface. In addition, I'm not sure what the flux is at their altitude. So you will get a lot of UV, but that won't be an issue inside the station. But I don't know if you get more cosmic rays. You might, even though that's inside the Van Allen radiation belts. So one of the problems that's taken place for many years is that in the process of shipping up items to orbital stations, people try to clean them carefully to make sure they're not carrying any microbes with them, which really amuses me in anything that's going to be human rated, because the humans are going to bring their own microbes with them. Right?
Absolutely.
Yeah.
So what they do here, and this is a wonderful paper for students because much of it is kind of old school. And I love the fact if you go to the isolation and cultivation bit, they actually have a specialized microbial sampling kit. And so what I like is the on-orbit sampling. They sampled before they launched some stuff up, and then they're on-orbit sampling, a predefined sampling location and procedures. And I like they have two negative control groups. One is they waved the wipes in the air for 30 seconds. That's control one. I want a movie of that. And then secondly, they actually don't take the second control out of the bagging. And that's a really good idea, right? And so they wiped different areas, and then they stored things at 4 degrees, and then they brought them back to Earth. And they did exactly what you might think. They put it into sterile water as an eluent, vortexed it. They even ultrasonicked it to enhance the extraction from the wipes. And then they cultured it in tryptic soy agar at 37 degrees for 3 days. And what they say here, and they don't give any numbers or anything, just talk about resulting bacterial colonies were isolated and purified. And
what kind of agar did they use?
It's probably nutrient agar.
Tryptic soy.
Okay. Yeah.
By the way, Mark, so they wiped before launch, is that right?
Yes.
And then up there.
Yes.
And I presume they wore gloves, right?
Yes.
Okay.
And my question is, who samples the gloves? I've always worried about that.
Well, sterile gloves are generally passed through in a radiator, and they're given many grays of radiation before they're certified as sterile. So hopefully they use sterile gloves.
So as far as I can see, I don't know whether they got a lot of things or they just picked likely what's the word I'm looking for? Likely suspects, as it were. And they got something that looked like bacillus, and that makes Petra happy. Right?
Well, they got a spore and
an endospore. Yeah.
And I was worried that the sonication would kill any vegetative microbes.
I don't know enough about it, but I know people have used that as a purification technique. And I like the way you think. I would have fully expected, and this is something we might hear in a subsequent episode of this podcast from the International Space Station, I would fully expect human-associated microbes everywhere up there.
Yeah.
Oh, yeah.
Just because we emit them all the time, leaving out the fact that we are by our nature sloppy eaters, and you have microgravity. So it turns out that what they ended up getting is a relative of bacillus, but it's actually closer to cytobacillus. And I like the way that they did this. They got full 16S. Now, this is important to come back to. They did full 16S from this, actually using techniques that I use. And it's funny because they actually found it to be closely related to this particular genus Nyalia, which I was not familiar with. But what's fun is you can actually see, and this is in figure 1, the kind of relationship between things. And Petra, how do you feel about the taxonomy of bacillus in general?
How do I feel about it? Does it make me emotional? Or the bacilli are a big group.
Yes.
And now they're called, probably one of you can pronounce this better than me. It's no longer called the firmicutes, the bigger group outside them. They're now the Bacilliota, I believe.
Yes. Bacilliota.
I'm always fascinated, though, because, when they found this story of Esther Ongert's, when they found this odd Epulopiscium, this giant bacterium that turned out to be in the Clostridia, the bigger group of bacteria. So I'm always excited to see another one.
Well, it's interesting. If you look at figure 1, you can see actually that the Nyalia appear to be from one event. They look monophyletic, and they are relatives more closely related to Cytobacillus and not that close to Bacillus. I also love the fact they use Panabacillus as their out.
Yes.
As their outcross. So that's a really good thing to do. Now, one of the things that I talk to students about a lot is the danger of 16S only. And so I'm delighted that they also did ANI, which stands for average nucleotide identity, as well. And I had not heard of this, digital DNA hybridization. And all that means is they make a prediction of the melting temperature between one strand from the target and one strand from the query. And that's, they used to have to do that, physically by measuring that process. And now they can do it with computers. And when you see that, it shows that really and truly there's some interesting stuff here. But yeah, again, it's in Nyalia, which is really interesting to me. And that's going to be in table 1. And so then they go kind of looking in the genome itself. They actually sequence the whole thing. Now, let's have a bet between all of us here. Do you really think they just got one type of thing out of this sampling?
Nope.
So the question occurs to me, why pick that one? And I don't have an answer for it. And
it sounds like the way they sampled and the way they processed, they were going to enrich for Bacillus spores anyways.
Yeah.
So
And I'm thinking it might be that now, of course, they didn't pasteurize, but the fact that they put it through an ultrasonicator or just a sonicator is probably probably the reason for it.
That seems like an unusual step to me. So.
Well, it's to extract the DNA because this is all sequence based. Right.
Yeah, exactly.
So in any event,
so that is a good point. So then if you I'm just working my way through this. So then actually you shouldn't have done anything to diversity then in that sonication step.
Yeah. So, I would say this is an isolate from the Chinese space station.
Yeah. Sure.
Yeah. And they're talking about the various things that are found here. They did the sequence, they annotated, they go through the usual levels of what kinds of genes are there. And they didn't find anything that was tremendously different, but they did find a couple of things. There's bacillithiol biosynthesis deacetylase. This is BSHB1. And that's something that's associated with combating oxidative stress and protein stability structures. So it turns out it's got 19 amino acid differences from the type strain. And that's interesting. They actually have a picture of this too. Ditto with SPLA, which is a transcriptional regulator for spore photoproduct lyase. And it's really good at dealing with UV damage. So if you look down at figure 3 and figure 4, they show you some of these differences. So they're not seeing that in some of the closer ones. For example, the Nyalia, which species name is Nielsen, and this is from some of Ken Nielsen's work where they were swabbing spacecraft before launching, right? And they're not seeing them there. One thing that I found very interesting, and I want your thoughts on this, is, they did the typical thing, temperature range, pH, everything looks pretty straightforward, except, and this is interesting to me, this strain uses gelatin. And as far as I can see, that's unique to it from the numbers of organisms they're looking at, the closest relatives. So that got me thinking, what does gelatinase work on? Collagen is one thing it works on. And that got me thinking about skin flakes, because they're not just skin flakes, let's be clear, they're moist skin flakes. Don't you think that would be a good, mini nutrient source?
Sure, yeah.
Especially if the skin flakes were mobilized by the stool extraction system on the space station, where they literally sit on a device that helps them evacuate and contain the stool within it. And it's really quite easy to allow for leakage, and the microbes will just go off and float around because the vacuum is pulling things out and trying to contain it.
And I don't think there's any good way to do that in microgravity other than to have a whole lot of extra space that they don't have up there.
They don't indeed. The other thing, Mark, is the way the astronauts take care of themselves in space. They don't have the luxury of showers. And so they're using wipes, to glorified baby wipes, if you will, to effectively sanitize themselves, that they can actually remain together without killing each other from their odors as their microbiome continuously changes back to the unwashed form of our microbiome.
So it's interesting. I'm staying away from the obvious jokes there, but it's interesting to note, and I found it interesting, they can tolerate sodium chloride, but don't like it very much. And most skin-associated microbes like the sodium chloride, right? Which I'm thinking about from sweat. But the gelatinase, gel-E, they actually say that is a key virulence factor facilitating biofilm formation. And you definitely would want to have a biofilm going on if you're in microgravity.
Here's another thing that we need to think about in space is down here on the planet, we're in an ambient humidity profile that is conducive to the microbes that are affiliated with us. But when you're in space, are they humidifying the air to a specific set point? Or is it like the desert, where you're just getting the moisture that the human race will expel as a consequence of breathing? And I don't know what the relative, and this is one of the things I wanted to see, is what is the relative humidity on the space station? Because that could influence the community of microbes that can actually thrive and persist on surfaces, because the great equalizer in the built environment is dehydration. That's effectively how alcohol hand sanitizers kill. They effectively dehydrate the microbe, and then they give up the ghost. And so in space, you may be shedding microbes, but because of the relative humidity, and which is another reason they may have only found spores, is because they basically made spores because there wasn't enough humidity for them to survive on the surfaces subsequent to their fugitive escape from the person who brought the microbe to the space station.
I think that the take-home lesson here is this is an isolate, and Petra, you'd be glad, white colonies, endospores, sizes are expected. That's nice to see, right? But if you're interested in adaptations to microgravity, I would recommend kind of having something you take up specifically to study that.
And watch how it changed.
Because you would have a beginning and you'd have an end. Because when we look at specific changes that have taken place in its genome, how many of those are associated with that environment and how many of them are associated just with random fixation of traits?
So, Mark, this isolate came from Earth, right?
Yeah.
We don't know on who or what, but it came and it's established itself on the space station, on the China station, right? That's the bottom line?
That's the bottom line.
Okay. But I agree. If you wanted to look at evolution, you should take up something that you know the genome and so forth, and then watch it evolve, right?
In addition, I don't believe this was a general survey. This is just an isolate.
Yeah.
And so it's what's not written that interests me almost as much as what was.
Yeah, I mean, essentially this boils down to they isolated one organism, essentially, and they did it in space instead of on Earth.
Mm-hmm.
I mean, and the humidity, okay, does the microbiome or the room microbiome change in the winter in the Northeast or even here versus the summer? But I mean, have we learned something about space from this isolate, or the fact that there are more bacteria in our guts than we know?
There are more
Well-
bacteria in our guts than we know, is I think there's the take-home message here.
And I'm not sure. I've got to go do a deep dive on this at another time, how much flux they're getting from cosmic radiation. They're not getting UV inside. We know that. And so as far as cosmic radiation, maybe it's more than we're used to at ground level, and that's important. But I don't think it's that much. I mean, it might be like Denver, right? Denver gets more cosmic radiation than sea level.
It might be like 5,000 feet. That's about as to.
Yeah. So I'm very interested in hearing about a real survey of what's there and how it changes over time. And hopefully that's being done.
I'd like to know, like, just somebody do a fluctuation test, just figure out what the mutagenesis rate is, something like that. That seems more, I mean, I'm guessing somebody did that. It'd be a little messy, but it would be interesting to know whether the mutation rate is higher with that more cosmic radiation. I don't know. I mean, it's at a low level here on Earth. I don't know what your Geiger counter does if you take a Geiger counter to space. Does it? Because all that background radiation is cosmic radiation for the most part. So
it might be grasping a little bit at microbial straws, grasping at pili. Anyway, something.
I don't know
like
if I
that.
could justify this in a grant.
Yeah, well, and I think that if because they actually wrote about things having to do with stress tolerance, and in fact comments about UV resistance, which I don't think is really relevant to this, unless they're somehow getting UV in on the inside, which I don't see how. It sounds. I would absolutely want to know if the mutation rate is different. Or if the repair mechanisms are
I'm
enhanced.
guessing somebody has actually done this on some level, but I don't know.
Sounds like a great undergraduate project.
Well, the other thing that we need to appreciate is that at least on our International Space Station, we use something called Whipple shields. And Whipple shields are effectively multi-layers of non-metallic material. And it's typically something really like Kevlar. To effectively absorb the radiation, deflect the radiation, direct the radiation. And it's generally anything that uses hydrogen-rich materials. When I was working with one of my colleagues who works for a private sector company, they were doing a lot of stuff with the International Space Station. And that's when I learned about Whipple shields. And it really helps reduce crew exposure to radiation. And that's where these experiments were done, in the crew's living space. They weren't done outside of the station. They were done in the shielded compartments. And I'll bet you that the Chinese space station is probably using something very analogous to Whipple shields, or they may have aluminum or potentially even lead, but I doubt lead because that's heavy.
Mark, thank you very much.
Certainly.
Loved it. Okay, now we have a story from Petra about radars.
About radar, although very much on Earth. I picked this actually for a couple of reasons. One is, though, the title just got my attention. I think when we write titles for papers, I'm not always great at doing this, but if we are putting our papers in journals, especially ones that might have people outside our field read them, I think it's really important to have titles which any person who might be flipping through this journal might understand. And they did a really good job, because this article, which was published in Cell recently, is entitled "A Chemical Radar Allows Bacteria to Detect and Kill Predators." So every word is clear and understandable, and you understand it's about bacteria and detection and killing, which is really what this article is about. This is by Xua Bing Zhang, Kevin Schlabach, Victor Hugo Perez Carrillo, Anand Ibrahim, Shahran Naim, Anna Komor, Ruchira Mukherjee, Somak Chowdhury, Lisa Reimer, Felix Trotman, Corinna Vlatt, Christian Hertweg, Ute Helmlich, and Pierre Stallforth. And many of these authors are at Leibniz Institute for Natural Product Research and Infection Biology in Jena, Germany. and others are at other institutes in Vienna. There's also University of Beirut, which is in Germany, this one, and Friedrich Schiller University, and also Goethe University in Frankfurt. So an international group, but at many different German institutions. And this paper caught my eye not only because of the title, but because it kind of goes to talk about this very complicated relationship that's going on, I think, in little conversations that are being had between microbes and the environment. And this is simplified. We're not talking about a microbiome. It's really a conversation between two kind of groups of organisms, the Pseudomonas syringae, which is a plant pathogen. It's a pretty good model system for people who study plant pathogenesis. And Polyspondylium pallidum, which is an amoeba. And this amoeba actually eats bacteria, and in doing so, it protects plants. And this paper kind of went out to kind of understand the molecular interplay, and they first kind of wanted to see if there are some of these bacteria, some strains that might be able to escape the amoeba. And this is in figure 1. And they start actually with a really nice, so there are a lot of pseudomonads out there. And the one that people are probably most familiar with, the TWiM listeners are most familiar with, is Pseudomonas aeruginosa, which is primarily a human pathogen, but it has actually, I think Fred Ausubel's lab showed many years ago, it can also be a plant pathogen. But this is Pseudomonas syringae. Putida is pretty common. You'll come across in fluorescence, also kind of big models, but this is syringae, and they're sort of a nice sort of all the pseudomonads in their relationship. And then we have a little tree, and you can see where these different isolates come in the tree. And so they have this isolates. They were isolated along with these amoebas, so they probably already have a relationship. They're kind of biasing towards that. And then they do what's kind of a plaque assay. So figure 1A is the tree. Figure B is a picture of the sort of the spores, the fruiting bodies of the amoeba. And then C is actually two of these strains of Pseudomonas syringae, SZ47 and SZ57. And they call SZ47 inedible, as in it doesn't get eaten, and SZ57 is edible. And these are a little difficult because there's this white background, which might just be a reflection. I'm not sure what it is. But if you look at the edible one, SZ57, they're doing basically a plaque assay, which is sort of a nice lawn of bacterial cells. And then they have holes where the amoeba have actually eaten them. So that's why they call them edible and inedible. And they want to figure out why these very closely related, you can see on in figure 1A, sort of isolates of Pseudomonas syringae, why they're being able to survive, or not get eaten. Survive, not get eaten, basically, same thing here. And they do some, I think, really kind of clever things because they're so closely related. They can actually look and see, first of all, what's being made by these guys. So they do some mass spec and NMR to kind of see which these two strains, 47 and 57, what they're making. And then actually they have the whole pathway mapped out here in figure 2, but figure 2A is they get what they call syringifactin, this one molecule that they see that's made by both of them, though. So both the one that's edible and inedible. And when they are in co-culture with the that's being made by the pseudomonads, syringifactin. But when these organisms are in co-culture with the amoeba, they get two more molecules coming up. They call them peptide 1 and peptide 2. Basically, though, those are derivatives of the syringifactin. So the syringifactin kind of helps the pseudomonas move. The amoeba eat the syringifactin and basically digest it in part. They deacylate it, delipidify it, and they spit out these two peptide molecules. Okay, so then the first thing that they do is with the syringifactin. Again, these are so closely related, they kind of go into the genome and they figure out the genes that make the syringifactin. So then they can make mutants that do that. But their real question is, okay, so they're making this thing that the amoeba eat. That's not that exciting. but they're wondering whether there's some difference between the two organisms and what is that difference. So they guess that maybe it's something about the syringofactin. Again, they feed it to the amoeba, and the amoeba make the deacylated peptide, the delipidified peptide that I told you guys about just a minute ago. And then they feed that deacylated peptide to Pseudomonas. And then they finally see a difference between these two strains. The one strain, the inedible one, the one that doesn't get eaten in the figure 1C, that guy actually converts it, this peptide, to something they call pyrofactin. And the other one doesn't really do much at all. It changes it a little bit. But when they feed the pyrofactin molecule to the amoebas die.
So the way I looked at this, Petra, is you can imagine that the syringin factor that the Pseudomonas syringae is making is a prodrug.
Exactly.
It lands in the amoeba transforms the drug, it sends it back to Pseudomonas, and then the Pseudomonas says, well, I'm going to return your serve and I'm going to really spike it.
Yeah.
And it sends back a lethal agent.
It's basically a pro-drug.
It's a pro-drug. You got it right.
It's a pro-drug, which is also amazing because what's happening here is the amoeba is unwittingly making a molecule that the Pseudomonads can turn into something that will kill the Pseudomonads.
It's a microbial tennis match.
It's a microbial tennis match, but instead of like just batting it back, they actually put spikes on it. And so the poor amoeba gets it back and it like blows up. I feel like it's something from a Roadrunner coyote.
Yes, it is.
It's a tennis game, but with a bomb.
Exactly. But
With
the
a
bomb gets
spike.
made as the tennis is being played. So many great analogies here.
Yes.
So I wanted to say something really quick, if I can, Petra. What I really like, most people when they hear about the amoebae, they'll say, well, I don't care about amoebae. Just bear with me for a second. And then I think, but that's what macrophages really are.
The macrophage, I kept thinking about that movie, that old movie of a macrophage, because these amoebae are protecting the plants. It turns out most of the Pseudomonas that screen get eaten by the amoeba.
And if you think about all the different techniques that disease-causing bacteria have to defend themselves against being used by a macrophage or eaten by a macrophage, this really factors in.
Yeah, I'd be curious if there's more of this. I think Pseudomonads are really good at making different things. So in a way, they're sort of the ideal organism among the gram-negatives for doing this kind of stuff where they're modifying. But at least in the gammas. But I think this is really, really cool. In any case, though, they want to know what's making this, which enzyme in 47, SCE47, is making the actual killing, the pyrofactin. And again, because these two strains are so closely related, and they know they know what the molecules look like because they've done all this mass spec and NMR, they go back and they look for enzymes that can make that. And they actually narrow it down pretty quickly. And they find a little gene cluster that they call CRA for chemical radar. One is called CRA-R, which turns out to be regulatory. And then there are two others, CRA-A and CRA-C, which actually do the modifications. And it turns out, and I thought this was, and they're necessary when they knock them out. They don't get the killing molecule, the pyrofactin. And when they put them into E. coli, they can have E. coli if they feed it the prodrug now, I guess. They can get E. coli to make the pyrofactin. So that was really cool. CRA-R is actually regulatory. It seems to be some kind of transcriptional activator. If they put the CRA-R into SZ57, they can essentially turn on the two. So it turns out that the CRA-C and the CRA-A are conserved, but they're off. You need this regulator to turn them on. So when you put the regulator into SC57, it becomes inedible. And there's actually this is actually one of the pictures in 1D. And I actually stared at it for a long time, and then I finally figured it out. So they're necessary. If you knock them out, you don't get it. And they're also sufficient. You can use E. coli if you give them the other thing. So I thought that was really quite nice. So now they have this little pathway and this regulatory set. Oh, the other thing that I thought was really interesting is we always talk about horizontal transfer. This feels like something horizontally transferred. The regulator, the activator, that seems to be some kind of weird recombination because they didn't find any homologs of it. They see CRA A and CRA C, the synthetic genes. They see those in all the pseudomonads, but the regulator, the on switch, is only in this one strain they isolated, which is wild.
Well, so it's like now we've gone from a tennis game to a doubles match, and CRA A is the doubles partner being added.
Yeah, but it's weird because it seems to have been some kind of, I don't know, you would think it would be more common.
Yeah, you would.
And, it's very, I thought that was kind of fascinating from a I've never heard of that
channeling
before.
John McEnroe right now. You must be kidding.
I'm getting a little lost in the tennis bomb kind of idea. But in any case, the thing needed to turn on the thing that makes this pyrofactin from the partially digested amoeba deacylated syringofactin. That seems to have probably come from 47's own genome somehow magically. And then the last kind of thing that they do, and I thought this was really quite lovely, because again, what's the point of this battle? It's actually not a tennis match for entertainment, although I'm very entertained at this point. It's actually a sort of battle between the amoeba and the syringe, this plant pathogen and the sort of Swiss guard, let's say, of the people who stand outside the U.S. Open, preventing you from getting in without a ticket, right? So, sorry to keep the tennis.
No, it is perfect, Petra. It's going to help the listeners.
In any case, the amoeba are constantly patrolling, eating bacteria. But when you have this ability to make the syringofactin and then have the amoeba digest it and give you back the pyrofactin, and so you can make the pyrofactin, then you actually infect the plants better. And that's sort of the last set of experiments, is putting it back. They do some really beautiful work, actually. I mean, there's just a lot of lovely little experiments. I'm kind of kicking myself for not picking this for our prelim.
Ooh.
Yeah, I didn't though, guys. But there's this beautiful, they're actually showing that the precursor, the thing that is going to actually be made into the pyrofactin, binds to this regulator CRA-R, and they can get some really beautiful spectra showing it bound. So they show that CRA-R is actually activated. So it's the sensor for this deacylated syringofactin. And so that's how you need the CRA-R, not only to turn it on, but to sense that the amoeba are there and eating, because why waste your time making this pyrofactin if there are no amoeba to kill out there? So, and that's figure 5.
It's hungry amoeba.
Exactly. So it really has something for everybody. It's got some really beautiful mass specs, some nice kind of evolutionary relationships, and then obviously this kind of chemical radar and match, and a little plant biology. So I love it. The figure 1 is great, and figure 6 is sort of the overview of sort of it going back and forth between P. syringae and P. pallidum, prey and the predator, and then I guess the prey becomes the predator again. So that's figure 6.
And it's open access.
And it's open access. It is, in fact, cell press open access specifically.
You may have said this, but the syringofactin, what's the purpose of the amoeba deacylating it? Would it be toxic to the amoeba otherwise?
I don't know if they...
No, I didn't see it, but I don't know.
I don't know if... It might just be that the amoeba... Yeah, I mean, I don't know what it looks like to the amoeba, the syringofactin. I think it helps the Pseudomonas. It might help the Pseudomonas motility or something like that, because it is in that class, but I don't know.
Is it a surfactant?
I think they said it did help the motility, but I could have missed it.
Because normally if you make a bacterium, and I hate this term, slippery, it causes trouble with phagocytosis.
Seriously?
Yeah.
Oh, yeah. That's why we have capsules on bacteria.
Yeah. That's why opsonization has to take place to get around that a little bit.
Yes.
So,
well, this syringofactin is...
Yep, they're for making the Pseudomonas swarms, and they're for swarming.
there you go.
I just want to know why...
Yeah, it is a surfactant, so like a little soapy, slippery bit.
Mm-hmm.
I just want to know why the amoeba are deacylating it, because that gets them into trouble.
But
Exactly.
it also prevents the Pseudomonas from swarming it removes the slipperiness of it. It's like decapsulating it.
It's only a very small subset that seem to have this particular CRA-R. They need the CRA-R in order to respond to it and to turn on the genes for making the pyrofactin.
I've read I'm not up to date on it, but I've read several papers about soil amoebae dealing with bacteria, and it really is a wonderful predator-prey story. And you've given us a new wrinkle on that.
Well, I'd like to thank the authors for giving us a new wrinkle. So it's really neat. And it kind of has something, each figure is different, has something for everybody. But it was just a new take on syringe. So.
And the figures are relatively easy to digest.
Yes.
They're not these overly...
Digest?
Yeah, well, digest. Normally, cell figures, you sort of have to spend a day on each panel.
They don't have more than like 4 panels.
Yes, they're digestible, as I said. No pun intended.
It's a nice paper. It's very compact.
Thank you, Petra.
Thank you.
All right, that's TWiM 331. You can find show notes at microbe.tv/TWiM. You can send us questions or comments to twim@microbe.tv. If you enjoy these discussions, we'd love to have your support so we can continue them. Microbe.tv/contribute. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin is at Washington University. Thank you, Petra.
Thank you.
And Mark Martin is at the University of Puget Sound. Thank you, Mark.
Always a pleasure to be here.
TWiM Adjacent. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.